I was looking to improve an osmosis lab I had done the year previously. Osmosis, if you remember from our osmosis video, involves water traveling through a semi-permeable membrane, essentially a cell membrane. And I wanted a cool way that students could model this in different scenarios. And one of my colleagues told me about this egg lab. I won't get into the whole lab though. It was actually one of the first steps of the procedure that got to me. To get ready for the lab, my colleague told me, you can soak eggs in vinegar for 24 to 48 hours and the shell comes off. Oh, so I need to make hard-boiled eggs then. No, raw eggs. But if the shell comes off, well, that's the whole point. What's underneath the shell is going to mimic a cell membrane. It's kind of modeling how a cell membrane would function. You know, if the whole egg was actually like a cell. So then you run different scenarios with it for your osmosis lab because it will be a semi-permeable membrane, like a cell membrane. I couldn't visualize this. If the shell comes off, but it's a raw egg, how does it stay together? So I have this area in my house that's designated for things for me to try out. Don't worry, I always clean up afterwards. I tried this experiment out in advance, just to be sure. The hard shell is removed, but the membrane that was always there remains. We often visualize the membrane of a cell this way, like this membrane around the chicken egg. The cell membrane is semi-permeable, meaning it lets some materials through, but not others. We have an entire video all about cell transport and how materials can pass through the membrane. A cell could never be as large as a single chicken egg, though. Why? Well, it turns out surface area is a very important thing. Remember that surface area determines the surface measurements of that cell membrane. And the cell membrane controls what goes in and out of cells. That includes food coming in, as well as molecules that are essential for metabolic processes, and then also waste going out. If volume, which is all this space inside, increases, then you will have more need for surface area, as you will have more need for food to enter, more of a need for waste to be removed, and more metabolic reactions occurring in this larger volume in the first place. If we do a little bit of math here between these two models, I'm going to use popular cube models instead of egg-shaped models because it's a little faster for me to do surface area and volume calculations. See how here there is a big difference in surface area to volume in the smaller model? 6-to-1 ratio. That means the surface area in the small model is 6 times more than the volume. Look at this beautiful ratio with so much surface area. But if we look at this bigger cube and we do some math for this model, that surface area to volume ratio decreases. Sure, the surface area is still larger than the volume in the large model, but it's only 2 times as large now, not 6 times as large.
In lab, some of you guys learned structures that increase cell surface area. What am I talking about? Microvilla, yes. Nice.
Cells are way smaller than this small model here to allow for an exceptionally large surface area to volume ratio. And a major reason why we're not going to find a cell as big as this chicken egg here. Surface area is important. And while we can model a lot of the processes of cell transport from this egg membrane and how important the membrane is, I don't want to neglect talking about the amazing cell membrane structure itself, because the cell membrane structure truly is magnificent.
What is this?
Phospholipid bilayer.
Phospholipid bilayer, right? Polar heads, non-polar tails.
And since every single living thing is made up of one or more cells, which is part of the cell theory, it's a big deal because every single cell has a membrane. It doesn't matter whether you're talking about bacteria, or protists, or plants, or animals, or fungi, even archaea aren't too cool to have a membrane. They all have a cell membrane. The structure can vary some, but we're going to be talking about some major structures of the membrane that you can actually find in most cells. We should mention that the fluid mosaic model is often what we use to describe the cell membrane. A mosaic, in case you've ever created one, we did in some of our art classes over time, arranges many small pieces together to make some large piece. You'll see that that makes sense when describing the membrane in a minute. The word fluid implies movement, and this is true for the cell membrane, as the components are floating around, they're not static. So let's take a look at some of these components in the cell membrane. We're looking first at a phospholipid bilayer. A phospholipid is a lipid, but an interesting one. So when you talk about a lipid in general, many lipids are nonpolar. Think of oil, for example. It's non-polar. It won't dissolve in water, water is polar. But a phospholipid is interesting because one part of it is polar, the head, and the other part of it is non-polar, the tail. It's amphiphilic. Let's explain what we mean. We often refer to the polar head of the phospholipid
Ringing bells, right? You guys all okay with this?
is hydrophilic, which means that part loves water. Well, you know, if a lipid could love. The non-polar tails are hydrophobic. They do not like water. These phospholipids arrange themselves into a phospholipid bilayer with the non-polar areas here in between, away from any water. It also allows this area in between to be separated from the outside and inside. Water can be found on the inside and outside areas. Also, these phospholipids, they don't just stay put. They move around. It's the fluid mosaic model after all. This gives the cell membrane flexibility. Phospholipids can even flip-flop around, but that's far less common. Remember that this entire phospholipid bilayer borders the whole cell. It would be a sphere, even though we're just looking at one area of it. We have an entire video that talks about which molecules can go in and out of this membrane and which ones can't that you can view, but for now, we're going to look at some of the other structures more in depth. Cholesterol. You know, cholesterol often gets a bad reputation. But while cholesterol that builds up in the arteries can be a problem, cholesterol in your cell membrane is critical. If temperatures drop, the cholesterol can actually function kind of like spacers between these phospholipids, keeping them from becoming too packed. Or vice versa, the cholesterol can actually function to connect phospholipids to keep them from becoming too fluid in warm temperatures. Protein.
Okay, remember we said cholesterol stabilizes cell membrane? We all good?
In protein synthesis, we talk about why it's so important for cells to make proteins. Many proteins are found in or on the cell membrane.
Okay, let's start doing notes now. Membrane lipids, we talked about phospholipid bilayer. Glycolipids we know about, right? What's a glycolipid?
Carbohydrates.
Yeah, it's a phospholipid with a carbohydrate hat. Got a little carbohydrate sticking up off of it. And cholesterol, we just saw again, helps stabilize the cell membrane. Now, there are lots of proteins in and around cell membranes. There are lots of proteins in and around cell membranes. And of course, they have different names depending on how they're categorized. There are peripheral proteins that are found on the outside of the cell. And there are integral proteins which are built into the cell membrane. I may have just confused you a little bit. I wasn't real tidy with my words. Peripheral proteins can either be inside or outside. Like this guy here would be a peripheral protein. You could also have a peripheral protein there. Either inside or outside. But what we're saying is it's not built into the plasma membrane. Peripheral proteins are not built into the plasma membrane. Integral proteins are built into the plasma membrane. Integral proteins are built into, actually part of, the plasma membrane. There's a special kind of integral protein called a transmembrane protein. There's a special kind of integral protein called a transmembrane protein. Take a wild guess and tell me which one of the ones up here are the transmembrane proteins. Yeah, that dude and that dude. They're transmembrane because they go the whole way through the membrane. Not all integral proteins go the whole way through the membrane. Transmembrane proteins do. Does that make sense to everybody? Let's see what the Amoeba sisters have to say.
And they play major roles. Peripheral proteins, like the name suggests, tend tend to be on the peripheral area of the membrane. So while they're on exterior areas of the membrane, they generally are not going to go through the membrane. That's where integral proteins. Integral proteins go through the membrane. Oh, and peripheral proteins can sit on them sometimes. Because of location, these proteins tend to have different functions. Integral proteins, with their potential to go through the membrane, are frequently involved in all kinds of transporting methods for all kinds of materials. Some relevance? Consider the breakfast you ate this morning. Your body digested what you ate for breakfast to obtain glucose. Once in the bloodstream, those glucose molecules can't just squeeze through the phospholipid bilayer to enter all of your cells. The glucose molecules are too big and polar. But your cells need glucose to survive, to make ATP, and they rely on integral proteins to get it. Peripheral proteins tend to be more loosely attached, since they're generally not stuck in the membrane. They have an assortment of functions such as acting as enzymes to speed up reactions, or attaching to cytoskeleton structures to help with cell shape. Both protein types can have carbohydrates bound to them, which can make them considered a glycoprotein.
Okay, so we had a glycolipid, there's also glycoproteins. Glycoproteins are proteins with sugar hats. How are we doing, everybody okay? All right, open up your books, page 64 to 65. Page 64 to 65 in your books, please. That, by the way, would be a transmembrane protein. It goes the whole way through. All right, now if you look at 64, 65, you can see there's integral proteins, there's peripheral proteins, glycoproteins, glycolipids. Together, they all are part of the cell membrane. Everybody got a pretty good picture of that? And remember, it's kind of sitting there floating like all ping pong balls in the bathtub. They're just kind of moving around. It's not solid, it's not locked in. So, functions of membrane proteins. These guys do a lot of stuff. Take a look at page 66. There's a listing of all of the membrane proteins. These guys are important. You should know these. Membrane proteins allow for transport. They help stuff move in or out of the cell. They help move stuff in or out of the cell. They can be either channels or carriers. They can be either channels or carriers. A channel is like a tube. Let stuff pass through or not. Let stuff pass through or not. A carrier is more like a turnstile. You know what a turnstile is? When you go through the amusement park or on the subway or something, there's a thing that click and let one person at a time go through. There's some kind of shape change going on to let stuff pass through the membrane. The channel is just a tube. The carrier, there's something else going on there that allows the material through the membrane. Proteins can also act as enzymes and catalyze reactions. These peripheral proteins here could very easily catalyze reactions inside or outside of the cell. They can catalyze reactions inside or outside of the cell. Proteins also act as receptors. If you look at page 66 on the second picture down, there's a receptor. You can have a protein sitting here, and it'll have a place where a hormone can bind to it. It'll have a place where a hormone can bind to it. That'll send a message into the cell. That'll send the message into the cell and say, hey cell, do this thing. Oftentimes, hormones are proteins, which means they're held together with polar covalent bonds, which means, can they dissolve through here? Can they dissolve straight through the cell membrane? No, because remember, this is non-polar in here. This is hydrophobic, hates water, hates anything that dissolves in water. So the protein, in this case it's a hormone, can't dissolve straight through. But it's got to get its message inside the cell. So if it sits in the receptor, this end of the protein will do something to say, hey cell, I found this hormone, we have to do our job. Does that make sense to you guys? Everybody's okay? If you have questions about it? This allows messages into the cell. Not the protein itself, but it gets a message in the cell, like a doorbell. Ding dong, somebody's outside. Ding dong, do this thing. You good? Intracellular adhesion. These proteins help cells stick to other cells. These proteins help cells stick to other cells. What would we look like if none of our cells stuck together? We look like a puzzle. Yeah. So all these cells stick to their neighbors, and they use proteins to do it. Cell-to-cell recognition. Cells have to recognize each other to make sure everybody belongs. Your immune system cells are constantly rolling through your body, checking everybody's ID card. Do you belong here? Do you belong here? Do you belong here? If they find somebody who doesn't belong, they kill them. The cell can do that with these proteins. And if you look on page 66, fourth one down, cell-to-cell recognition, that protein is checking out the carbohydrates on the outside of the other cell's protein. It's making sure it belongs here. We talk about carbohydrates, cell-to-cell recognition, identifiers. And finally, proteins help attach to the cytoskeleton and extracellular matrix. Proteins help attach to the cytoskeleton and extracellular matrix. Remember, cytoskeleton is like a whole bunch of wires or ropes inside the cell. Gives the cell its shape, lets the cell move around. Extracellular matrix is the material outside of the cell. Extracellular matrix is the material outside of the cell. What's going on outside? So it helps give the cell its shape. It also helps the cell stick where it wants to be. Helps the cell hang on to the material outside so it's not moving around too much. Does that make sense to everybody? Any questions about those functions? All right.
If the carbohydrates attach to a phospholipid, you can have what's called a glycolipid. Glycoproteins and glycolipids can identify the cell as belonging to the organism, self and non-self recognition, which is very important when you're fighting pathogens. They can also be involved in many kinds of cell signaling. In fact, here's some relevance. A glycoprotein known as CD4 is found on the surface of some of your immune cells. The CD4 glycoprotein is essential for some of these immune system cells to interact with each other and activate. However, it's also exploited by the HIV virus. The HIV virus uses that CD4 glycoprotein as a way to bind to helper T cells, which it then can infect. Understanding the components of the cell membrane and how those components are involved in recognition and cell signaling is critical to understanding how to fight back against.
All right. Does that make sense to you guys? You guys are okay? You kinda like the Amoeba Sisters now? It's not a bad tool, again, not a bad tool at all to use. Membrane carbohydrates, glycocalyx. This is the phospholipids, glycolipids and glycoproteins. Glycolipids and glycoproteins. They make up the glycocalyx. That's the sugar candy coating on the outside of the cell. And again, that's there to help ID the cell. To help the cell, hey look, I belong here. Glycocalyx is used in cell-to-cell recognition. Now we're doing some pretty funky stuff medically. One of the things we're doing is we are genetically engineering pigs. They have the same glycocalyx as humans do. If you take a pig heart out of a pig and put it in a human, it's going to get rejected immediately. Oh my God, this is a horrible thing. Get it out. Danger, intrusion. But we're changing the genes of a pig. So the outside, their glycocalyx in their heart matches up with human glycocalyx. So we'll be able to transplant pig hearts into humans. Because I'm sure you guys are probably aware, we could use more hearts for transplant, right? There's lots of people waiting in line for organs, hearts especially. It's hard to come up with a heart somebody's not using. So we need more hearts. What do you guys think about that? Do you think, first of all, is that medically ethical? We're gonna raise pigs for human parts? Are you guys okay with that? Do you eat bacon? Because you raise pigs for bacon. That's what, I mean, if you're vegetarian, cool. But if you're eating ribs or pulled pork or bacon, you know, why not make human hearts?
Do you think that path is better than growing your own heart again through stem cells?
Probably not.
Yeah.
But it's still, it's under research right now. Yeah, it's under research right now. And if you need one pretty quick, and there's a body there. Can anybody see other ethical concerns? Anybody see problems or maybe dangerous areas?
I might not like bacon.
Might not like bacon anymore, you'd be kind of an animal. Yep.
Do you think they would be more susceptible to like diseases that pigs?
Absolutely, that is one of the concerns. If we start putting pig tissue in people, we may become more susceptible to diseases that right now only infect pigs. If we allow that window to jump species, the weird stuff can start happening. We can start getting some more critters like the coronavirus. How many guys are flipping out about the coronavirus?
We're all young. So.
Yeah, you guys are fine. Don't worry about it. Did I talk about this with you guys, or was it the other class? Okay, coronavirus. You got about a 1 in 1,000 chance of dying. Mortality rate's 0.1%. It's only going to hit you if it's more likely to kill you if you're old, very old, or very young. So you guys are probably okay. The flu kills as many people as the coronavirus does. Just everybody's, oh my God, coronavirus. Well, the flu's here. The flu's already killed thousands of people in the United States this flu season. They've all died of flu. I forget what the exact number is, so I'm not going to say it, but I'm pretty sure it's in the tens of thousands. So coronavirus isn't really that big a deal. Ooh, scary. Now, if we get something that's instead of a 0.1% mortality rate, if we get something that's 10% mortality rate, 1 in 10 people get it and die, look around the room. There's probably 30 people in here, so 3 people would die from it. Now imagine 3 people dying everywhere you go. That's fierce. Our healthcare system would not be able to handle something like that. The hospitals would be jammed, there would be no room for anybody. Society would break down, holy crap. I don't want to get into the whole doomsday thing, but that's a potential that could happen. So always wash your hands and get your vaccines, right? Do it. Anyway, glycocalyx pigs. Did anybody see some religious concerns using pig hearts? If you're Jewish or Muslim, do you think you're gonna take a pig heart? Probably not. Up to you. If it's your kid that needs a heart, that's gonna be a really interesting ethical dilemma, isn't it? Yeah. So we have some interesting things coming down the road. Now the next question is, if you really need a heart, and there's a pig heart available, do you think you're gonna be able to afford it?
Yes.
No, you're not. Because the insurance company sure isn't gonna pay for it, and none of us have our own jets. So, you don't have enough money to do that. So there are ethical concerns, there are some interesting things coming down the line. Genetically engineered pigs are kind of an offshoot, there's other stuff happening in other places that we'll get to sometime this semester. I don't know if I wanna talk about this or not yet, but might as well, we're having a little conversation. You guys know that in China, they have genetically engineered human beings. They have changed the DNA of human beings. 2 twin girls that they added some DNA to. The reason for the experiment was to prevent them from having HIV. It turns out one of their parents, or maybe both, I forget which, had HIV. So they decided they were gonna change the girl's DNA so they could never get HIV. Okay, cool. But the change that they did, it's called chemokine receptor number 5, they made a bunch more of them, they mutated, it doesn't matter. The change they did also may make them smarter. So they're genetically engineering kids to probably be smarter. What do you guys think about that?
I think genetic engineering is gonna be the next frontier, man. How are we gonna live in space? How are we
Oh,
gonna?
it is, yeah, it is already. There's people doing stuff in their garages and their basements that is pretty crazy. So we might as well do the whole thing, we might as well blow another 20 minutes on this. In the next 20 years or so, you guys may be able to go to a hospital and have in vitro fertilization, test your baby, and be able to check a box that I don't want my baby to have any genetic diseases. How many of you guys would do that? How many of you guys would have a baby that was guaranteed it would not have any genetic disease? Something to think about, especially if you know somebody who has a baby with Down syndrome or one of the other things that you could prevent with this. Now, if you're in the hospital, there's also 2 boxes there that I want my baby to be cute, and I want my baby to be strong and athletic, and I want my baby to be smart. How many guys would check those boxes? You want a good-looking, strong, smart baby? How many would love your baby anyway, no matter if it was goofy and dopey looking and all that sort of stuff? Yeah? Cool. Good. Now, ask yourself this. How's your goofy looking, kind of not real smart baby gonna compete against the checkbox boop, 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 boop babies for jobs and other things? Your baby's gonna be, there's nothing wrong with this, your baby's gonna be flipping burgers and digging ditches. And that baby's gonna be superstar. Now, another question, can you afford that? No, you don't have your own jet. That's what's coming down the road, go ahead.
So my concern with that is just like the opioids, they said they wouldn't hurt anybody,
Yeah,
so.
that's an awesome concern because
Years down the road, you don't know what else is gonna go wrong.
that's an awesome concern because there are things coming out of China that maybe those 2 girls there are issues with. There are problems. And most countries aren't real forthcoming with their failures. Both US and China and everybody else is gonna say, oh, we really screwed up here, everybody look at this. We're gonna say, oh, nothing to see here, move along, citizens, move along, citizens. But there's already some information coming out that those girls have problems. So not only are we genetically engineering people and messing them up with our failed attempts at genetic engineering. And to make things even more interesting, also in China, they're adding genes to monkeys to make them smarter. Do you want Planet of the Apes? Because that's how you get Planet of the Apes. It's happening now. I mean, you guys are on the edge of this stuff. I'm gonna be dead by the time that stuff happens. You guys are the ones that need to be paying attention to what's going on around you. Go vote, guys, vote. All right? Because we there and do nothing, weird stuff's gonna happen. All right, enough politics, enough stuff, let's get back to work. Everybody okay with membrane proteins and membrane carbohydrates? Any questions about those? All right, specializations of the plasma membrane, microvilli, page 67, those are finger-like projections of the plasma membrane, and you already knew that, right? Finger-like projections of the plasma membrane that increase surface area. Finger-like projections of the plasma membrane that increase surface area. You might have a lot of microvilli cells in the small intestine because you gotta absorb lunch. Your stomach begins to break down, your small intestine does most of the digestion. The small intestine absorbs almost all the food you eat. But microvilli take more surface area, so you have more proteins that you can absorb more nutrients. Does that make sense to everybody? Membrane junctions, also on page 67. We have to stick cells together. Several ways to do it. Tight junctions are zipper-like series of proteins. They look kind of like a zipper that form waterproof junctions between cells. They stick cells together with a waterproof seal. Stick cells together with a waterproof seal. Where in your body would it be nice to have a waterproof seal?
Bladder.
Yeah, your bladder. You do not want your bladder to leak. You don't want your stomach to leak either. You don't want your skin to leak either. Tight junctions all over the place in your body.
You guys.
Anchoring junctions. Anchoring junctions are also called desmosomes. Make sure you know the word desmosomes. Desmosomes are real strong connections between cells. Desmosomes are really strong connections between cells. They're a lot like spot welds, if you know what those are. Look at the back of your washing machine. But they're strong connections between cells. Cells that might have a lot of desmosomes would be cells like, again, your skin. You don't want your skin to rip or shred, right? Also, your cardiac muscle cells, your heart muscle cells, are stuck together with desmosomes. It's a bad day if your heart blows up, right? So, cardiac muscle has lots of desmosomes as well. And then there are gap junctions. Gap junctions allow for communication between cells. Gap junctions allow for communication between cells. Allow cells to talk to each other. An example of that would be cardiac muscle again. All your cardiac muscle cells have to beat together. So they use gap junctions to talk to each other. There's a wave of calcium ion that flows through all your cardiac muscles. So your heart beats as one unit. Gap junctions are the passageways that calcium goes through. Does that make sense to everybody? All right. Now, moving to materials across plasma membranes. First thing we need to know, plasma membranes are selectively permeable. What does that mean, selectively permeable?
Don't allow certain things.
Yeah, it allows some stuff through and not others. It allows some stuff through, not others. Now we need to talk about ways things move in and out of the cell. There passive processes and active processes. Passive processes don't require any energy. Passive processes don't require any energy to move in and out of the cell. There's no ATP used. Active processes use ATP. Active processes use ATP. It takes energy to do an active process. Is everybody okay with that? Now we got to talk about a concentration gradient. A concentration gradient describes how much of a chemical is in one place versus how much of a chemical is in another place. Right now, we have a pretty even distribution of students through the classroom. There's not one particular area here that has a whole lot of students and one that doesn't, right? You with me? Now, if we got all you guys moved over to one corner, then the students would be really packed together, right? There would be a concentration gradient. There'd be an area of high concentration and an area of low concentration. Does that make sense to you guys? I'm going to try a little experiment. I'm just doing this off the top of my head, totally winging it. May turn out okay, may not. Do you want to try to do it? Okay, I'm going to multitask here. Usually, towards the beginning of the semester, I talk to my students about what happens if we get an active shooter drill on campus, right? Stuff, so we have to talk about it, but we got to kind of talk about it. My contract with you, if we get some kind of weird alarms going off, you all heard the alarms?
Yeah.
Like they're all through campus, they're in all the rooms, and you know, blah, 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 all that sort of stuff. If we hear something, I'll go outside and I'll lock that door, and I'll come back in. And then we'll be in the locker room, and we'll be okay. If that happens, what I want you guys to do is all move to back here. Come on with me, get out of your seats, come on. All move to some place where you can't see you if somebody looks in the window. There's windows there, there's a window there. Get someplace where they can't see us moving in. All right? So, if that happens, and somebody comes through the door, I don't care who it is, what do we do to them? We jump at them, we throw books at them, we hit them with chairs, we kick their ass. All right? Because if this happens, we're going down with a fight. Not, oh no, stop. Everybody with me?
Yeah.
All right, now let's look where we're at. We got a high concentration gradient of students against the wall, and a low concentration gradient down there, right?
Mm-hmm.
Make sense? All right, go back to your chairs. Or let's say this, diffuse. Back to your chairs. This is diffusion. Stuff is moving from a high concentration gradient to a low concentration gradient. That worked out surprisingly well. Considering I just now made it up. So that's diffusion. Moving from a high concentration gradient to a low concentration gradient. And it's a passive process. Doesn't take any ATP to do it. Usually what I would do is talk about this. I used to do it, but I didn't stop doing it. If I had some perfume, and I sprayed it right here, you would smell it first. You guys close. But it would spread through the whole room, right? Moving from an area of high concentration to an area of low concentration. That's diffusion. Diffusion moves down the concentration gradient from an area of high concentration to an area of low concentration. How are we doing? Are we okay? Passive process, diffusion. That would be simple diffusion. Simple diffusion doesn't need any help from anything. Simple diffusion is perfume spread through a room. Simple diffusion, if you put a cube of sugar into a cup of coffee and let it sit for a while, the sugar's gonna move all through the coffee. Now it'll leave some sludge on the bottom, but you get the idea, the coffee's gonna be diffused. Because it's gonna diffuse through the coffee. That's simple diffusion. Facilitated diffusion means there are carriers or channels involved. Facilitated diffusion means there are carriers or channels involved. Now, if we look back over here, this is like a channel. So I can have something with a high concentration move to an area of low concentration through the channel. Still doesn't take any energy because I'm moving from high concentration to low concentration. But I need that channel there. I need that pathway for it to get through. That's facilitated diffusion. Does that make sense? It works with a channel, or it works with a carrier too. If I had a turnstile or an atom there, a molecule there, that kind of did this and let stuff through one at a time. That's still facilitated diffusion. It needs help. It still goes from high concentration to low concentration. No ATP. How do we feel about that? Okay? Osmosis. This is important. Osmosis is the diffusion of water across a semipermeable membrane. Osmosis is the diffusion of water across a semipermeable membrane. In your book, on page 71, there is a classic experiment. There's a U-shaped glass tube. Looks like this. So if I pour water in this side of the U, what does it do? It goes down to the bottom and it'll fill up both sides equally, right? Everybody's good with that? Now let me put a semi-permeable membrane here. That membrane will let water through it like a stream. If I pour water in this side, is it going to fill up and be equal? Yeah, sure. So let's do that. So there's water, same level on both sides of the tube. Water is moving freely across the semi-permeable membrane. So if I tilt it like this, water's going to run over here. If I tilt it like this, water's going to run over there. It moves freely. Everybody with me? Now, let's put some salt in there. We're going to put salt on one side. And here are the sodium and chloride ions. Now salt can't go through the semi-permeable membrane. So I put a bunch of salt in that one side. Water is going to move from a higher concentration of water to a lower concentration of water. It's going to move down the concentration gradient. Which way is it going to go? Is it going to go to here or is it going to go over there? Yeah, water's going to move towards the salt. Water's going to move this way because there's more water over here. The salt's taking up room over here. Imagine it this way. You're out cutting the grass in August. Come back in, you're sweaty and thirsty. I got 2 glasses of water. One is just water. The other is a whole bunch of marbles and water. Which glass do you want? You only get one. You want the one without the marbles, right? Because there's more water there. There's more water here. Salt's taking up space. So, more water, less water, water's gonna flow this way. Salt's stuck there, it can't move across. Eventually, you're gonna end up with more water on this side. Because water, it's moving both ways, but a lot more is moving this way. Water moves from here to here. This side fills up. Does that make sense to you guys? This is called osmotic pressure. Something's pushing on this to move the water this way. That thing is osmotic pressure. Imagine it like this. Salt, this isn't really the way it works, but we'll try it. Salt is like a sponge. It's going to suck up water. So it sucks the water over to the other side because there's more salt dissolved. Does that help at all? Water's going to move towards the saltier side because it wants to be, it moves where there's less water. More salt equals less water. You kind of okay? Anybody have questions about this now? Because it's going to get worse in about 10 seconds.
Well, salt absorbs water, right?
Salt water. Salt water is going to, if you put salt water next to fresh water, it's going to suck the fresh water into it. Because there's more dissolved salt, less water. Alright, these are words you guys need. Hypertonic, hypotonic, and isotonic. Tonicity is the measure of how much material is dissolved in water. Tonicity is the measure of how much material is dissolved in water. I have a beaker of water. I have a red blood cell. I'm going to put that red blood cell into the water. Y'all with me? Got a red blood cell in water. Something you need to know. Inside of that red blood cell is equivalent to 0.9% sodium chloride. We're kind of salty on the inside. We're equal to 0.9% salt in water. We're a little salty. Inside the red blood cell is always 0.9% sodium chloride. If I put that red blood cell in water that's also 0.9% sodium chloride, I have an isotonic solution. That is an isotonic solution. The dissolved stuff on the inside equals the dissolved stuff on the outside. The amount of stuff dissolved on the inside is equal to the amount of stuff dissolved on the outside. What's going to happen to that red blood cell? Nothing. It's going to be fine. Water's going to move in and out equally. Red blood cell will be fine. Life is good. Now let's take another beaker. Put a red blood cell in it. Inside of that red blood cell is still 0.9% sodium chloride. But let's put it in at 10% Sodium chloride solution. Really salty. Really, really salty. You wouldn't want to drink that. That'd be like, holy crap, that's salty. What's going to happen to the water? Or better question, what's going to happen to the red blood cell? It's going to shrink. Because all the water moves out into the salty beaker. All the water in that red blood cell moves out into the salty beaker. That red blood cell is going to go like that. It's going to shrink. By the way, there's a fancy word for that you guys need. It's called crenate. C-R-E-N-A-T-E, crenation. Crenation is when the red blood cell shrinks because you put it into a hypertonic environment. Put it into a hypertonic environment. That red blood cell is going to shrink. Now, if we do the other thing, another beaker of water, another red blood cell, and this time we put it in 0.001% sodium chloride. Hardly any salt at all, almost pure water. You can do pure water too. Almost pure water. What's going to happen to that red blood cell? It's going to blow up and pop. Because all the water is going to want to be inside the red blood cell. That's called a hypotonic environment. Red blood cell will blow up, and if that difference is enough, they'll pop. You can actually watch that happen if you get a microscope and put blood under a slide, put a drop of distilled water along the edge of the slide, and poof, you just disappear in a little puff because they blow up and pop. So all the water moves inside. Isotonic, hypertonic, and hypotonic solution. Iso is the same. Hypertonic means it's saltier outside. Hypotonic means it's not as salty outside.
Is there a term we need to know for it blowing up?
Plasmolysis. 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 That's an old term way back in the Dark Ages. They used to call it plasmolysis. That's not. You won't find that. Is everybody okay with this? So if you're hanging an IV bag in a hospital, which one of these do you want to do?
Isotonic.
Isotonic. Remember, guys, work in a place where they hang IV bags. If you look, it'll say 0.9% sodium chloride on it, or 5% dextrose glucose. Those are isotonic. They're not going to make the blood cells do anything crazy. Now, sometimes they'll get off of that, but you have to call a pharmacy and do all kinds of funky stuff and lots of tests. Don't mess around. Isotonic solution is good. Hyper and hypo can cause problems. Everybody okay? All right. Oh, by the way, sorry you guys know what happens for all these guys. Filtration. When you filter something, you need the other kind of pressure. We talked about osmotic pressure. When you filter something, you have to have hydrostatic pressure. Hydrostatic pressure is just from pushing on a fluid. With gravity, it can be a pump that causes hydrostatic pressure. How many of you have ever put your thumb over a garden hose? Squirts a lot, right? That pressure, that push, is hydrostatic pressure. That pressure is coming from a water tower, or or maybe a pump at a substation, but that's hydrostatic pressure. Where in your body would you find hydrostatic pressure?
In your arteries?
Yeah, in your arteries, blood vessels. And the pump is heart. Yeah, that pushes stuff. You use hydrostatic pressure in your kidneys to filter out waste. Active processes. Okay, the amoeba sections are a little too long. This is way long. But crash course biology, watch it on your own time. This is 3 minutes and 50 seconds, let's try it. Active transport. Oh no. Is that obnoxiously loud? You guys okay?
Recall that there are
Helps if we turn this on, doesn't it? Everybody doing okay? Any questions? Oh, by the way, the fellow who did that experiment in China, altering the twins, now he's in trouble. They say, you did a bad thing before it went crazy. They're like, oh, well, this is cool, look at us. It's kind of fun to read about in a creepy sort of way.
two types of cellular transport, passive and active. Passive requires no added energy, but active does require added energy. Active transport also moves from an area of low concentration to an area of high concentration, or up the concentration gradient.
That's important, write that down. Everything we've been talking about so far goes down the concentration gradient. Moves from an area of high concentration to an area of low concentration. All passive processes move from high to low. Active processes move from low to high. Active processes move from low concentration to high concentration. Your body is not going to use energy for no reason. It has to use energy for these processes because you're moving against the concentration gradient. Otherwise, you wouldn't spend the energy. You just let it happen naturally. Active transport uses ATP, goes against the concentration gradient from low to high.
This is the opposite of passive transport, and because it's more difficult, it needs the energy molecule called ATP, which stands for adenosine triphosphate, because there's an adenosine molecule and 3 phosphate molecules. There are 3 types of active transport: membrane pumps, endocytosis, and exocytosis. Let's look at membrane pumps first. Membrane pumps are just carrier proteins that move substances from areas of low concentration to areas of high concentration.
Okay. Take a look at page 72 real quick in your book. There are some fancy pictures of red blood cells we just talked about, isotonic, hypertonic, and hypotonic. You should take a look at that. And then page 74. That is the sodium-potassium pump. Sodium-potassium pump. That is the classic example of active transport. Sodium-potassium pump is really, really, really important when you do things like contract your muscles and use your brain. Sodium-potassium pump pumps sodium and potassium. You should know it pumps 3 sodiums out of the cell, and 2 potassiums into the cell. Pumps 3 sodiums out of the cell, 2 potassiums into the cell. You need those numbers. I don't ask you to learn a lot of numbers, you need those. 3 sodiums out, 2 potassiums in. This is an active process, so you're moving against the concentration gradient. There's already lots of sodium outside the cell. We are throwing more out. You're moving more out against the concentration gradient.
Let's look at the sodium-potassium pump to see how it works. Outside the cell, there's a high concentration of sodium ion, or Na+. The brackets represent the word concentration. Inside the cell, there's a high concentration of potassium ion, or K+. The cell has ion channels that will allow these ions to diffuse and reach equilibrium, but the cell doesn't actually want that. It wants to keep a difference in concentration. So the sodium-potassium pump will force more sodium out and more potassium in all the time. Let's see how this works. First, 3 sodium ions from the cytosol bind to the protein, and a phosphate group from ATP breaks off in the carrier protein. This provides energy for...
Remember how we talked about breaking off one phosphate group? You just saw it. You want to see it again?
phosphate group from ATP.
Boom, there we got the ATP. Got 1 phosphate in the little binding site. There's the other 2 phosphates.
Breaks off.
There it goes. Leave behind the phosphate. This is done now. Your metabolism is going to be all about sticking a P back on there to reuse what's now ADP. You guys know what ADP is? There's a diphosphate. There's 2 phosphates. ATP, there's 3. Break the phosphate off. Now you got ADP. Stick it back on. You got ATP again. You can do this again.
Off in the carrier protein. This provides energy for the shape of the protein to change, and the sodium ions can be released into the outside of the cell. Now 2 potassium ions will enter the pump, which causes the phosphate to break off and return the pump to its original shape and release the potassium into the cytosol. Again, sodium will diffuse into the cell on its own, and potassium will diffuse out. The pump forces the sodium to be outside the cell and the potassium to be inside the cell. Another way that cells do active transport is through...
You guys okay with sodium potassium pump? Move this up against the concentration gradient, takes ATP, 3 sodiums out, 2 potassiums in. Tons and tons of videos. Probably get one to watch it. Actually, the Amoeba Sisters transport is pretty good, and so is Crash Course transport.
vesicle movement, either endocytosis or exocytosis.
What's a vesicule? Yes, it is a membrane-bound, you call it an organ, oh yeah, it's a bag of stuff. We won't use bag of stuff on a quiz or anything like that, but it's a bag of stuff. It's a membrane surrounding something you want to have in a container. It's a bag of stuff. Endocytosis and exocytosis. Which direction do you think endocytosis goes?
Inside.
Into the cell. And exocytosis? Out of the cell.
Cytosis brings items into the cell, and exocytosis pushes them out of the cell. Endocytosis is the process by which cells ingest external fluid, macromolecules, or other large particles, even other cells. We can be more specific about what materials undergo endocytosis. Phagocytosis is where large particles or whole cells are ingested, and pinocytosis is when solutes or fluids are ingested.
Okay, let me give you a couple words. Under endocytosis in your notes, you see it says phagocytosis. Think about phagocytosis like cell eating. Phagocytosis is like cell eating. You guys all remember Pac-Man, right? And phagocytosis. Now, in your notes, you'll see it says bulk phase endocytosis. Beside that, I want you to write pinocytosis, like it is up there on the screen. Beside bulk phase endocytosis in your notes, I want you to write pinocytosis. They're the same thing. I'm going to use pinocytosis. I'm not going to use bulk phase endocytosis. I'm going to use pinocytosis. Phagocytosis is cell eating. Pinocytosis is cell drinking. Cell takes in fluid and some protein from the outside and moves it inside. So cell eating is big chunk, maybe bacteria or big chunks of other stuff. Cell drinking is a little sip of fluid, like that. Cell eating, cell drinking. Phagocytosis, large particles. Pinocytosis, mostly fluids.
You can think of phagocytosis as cell eating and pinocytosis as cell drinking. So you can see the way it works. First, the cell membrane begins to pinch around the substances, and eventually it forms a vesicle, which can carry the substance to any part of the cell. Exocytosis is basically the opposite of endocytosis. It's a process by which a substance is released from a cell through a vesicle that transports it to the cell surface and fuses with the cell membrane. A vesicle can merge with the...
So exocytosis is basically taking out the trash. Or taking out protein that the cell made and wants to release. It's not necessarily all trash, but it's moving stuff from inside the cell to outside the cell. It's kind of the opposite of phagocytosis. It's taking stuff in, it releases it to the outside of the cell. One more term there real quick I want to get to. I know. Receptor-mediated endocytosis, that means receptors catch whatever it is you want to have. Kind of like fishing, the receptors catch it, and the receptors are all pulled inside the cell. The receptors containing your targets are all pulled inside the cell. And if you look at page 77, there you'll see all three of them. There you'll see all three of them. Everybody okay? Slide be free.